Perfect. Today, I'll be presenting a 2020 study by Qian, Zhang, and Park, published in Molecular Biology and Evolution. This study examines whether phylotranscriptomics, using RNA-derived sequences, is as reliable as phylogenomics, which uses whole genome data. This question matters because genome sequencing can be costly, and transcriptome data is cheaper and more accessible. In evolutionary biology, constructing phylogenetic trees helps us understand species relationships. Genome-based phylogenetics, or phylogenomics, uses massive amounts of data but at a high cost. Phylotranscriptomics is appealing because transcriptome data is cheaper and easier to obtain. However, it is not clear if transcriptome data, which only includes expressed genes, provides the same accuracy. Issues like incomplete gene representation and tissue-specific expression raises questions about its reliability. The researchers aim to directly compare trees built from genome data with those from transcriptome data. They wanted to see if the tissue source or the method of identifying orthologs affected the tree's accuracy. Importantly, they tested this across both mammals and plants to check for consistency. They selected 22 mammal species and 15 vascular plants, all with available genome and multi-tissue RNA-seq data. This allowed direct comparison between the genome-based and transcriptome-based phylogenies using real-world data. A critical step is identifying orthologous genes, genes that are inherited from a common ancestor. They tested two approaches: hamster, a commonly used method that's less strict, and the YS method, which uses gene trees for stricter ortholog detection. This comparison allowed them to assess how ortholog detection affects the accuracy of phylogenetic trees. Once orthologs were identified, they used maximum likelihood methods to build trees, a standard approach in phylogenetics. They compared the resulting phylogenomic, PG trees, with phylotranscriptomic, PT trees, using a metric called topological distance, which measures how similar the tree structures are. For mammals, the PT trees built with the YS method closely matched the genome-based trees. However, trees built with hamster had noticeable inaccuracies. Interestingly, using transcriptomes from different tissues didn't hurt the results much, and even mixing tissues among species only slightly reduced the accuracy, which is good news for practical studies. In plants, the pattern held true. YS-based PT trees align closely with PG trees. This supports the idea that phylotranscriptomics can be a reliable method beyond just mammals, making it a promising approach across diverse taxa. The study is robust because it used large data sets, tested across two major taxonomic groups, and compared different analytical methods. They didn't just test one condition. They examined multiple variables, which strengthens their conclusions. However, there are limits. They only studied mammals and vascular plants. More groups need to be tested. Also, the success of phylotranscriptomics depends on using strict ortholog detection. Plus, increasing the number of species in a study might reduce the pool of shared orthologs, which could affect results. The main takeaway is that phylotranscriptomics can match the accuracy of phylogenomics when rigorous ortholog detection methods are used. This suggests researchers can use transcriptome data reliably, especially when full genome data isn't available or affordable. This finding opens doors for more widespread use of transcriptome data in evolutionary studies. It also emphasizes the importance of choosing the right computational tools, especially for ortholog detection, to ensure accurate results. Going forward, researchers need to test these findings in other groups like fungi, invertebrates, or microbes. Also, it's important to balance the number of species sampled with the quality of data used. Overall, this study gives us confidence that with careful methods, phylotranscriptomics is a powerful and practical tool for evolutionary biology. Thank you. Have a great day.